My name is Emma Wallace, and I am a master's student in North Carolina State University's Department of Plant Pathology. The topic of this video is the mode of infection and disease cycle of Pseudoperonospora cubensis, the causal agent of cucurbit downy mildew. This is an important topic because cucurbit downy mildew is an economically significant disease not only in North Carolina but across the United States. Knowing the pathogen's biology and the steps of its disease cycle help in the development of control strategies and provide fundamental information that guide further research of the pathogen. This project illustrates the mode of infection and crucial parts of the disease cycle through basic stop-motion animation of clay figures, aka claymation. I molded host and pathogen structures and photographed the infection process frame by frame. I used Microsoft PowerPoint to add labels to some of the frames and Microsoft Movie Maker to put all the frames together and combine audio commentary. When trying to understand the pathogen and the disease it causes, it is useful to see the host-pathogen interaction clearly illustrated. I hope this animation aids in your understanding of P. cubensis and cucurbit downy mildew. Pseudoperonospora cubensis is the oomycete pathogen that causes downy mildew on cucurbit leaves. This pathogen causes chlorotic lesions to form on the leaves, mostly in areas near vein junctions. The shape and size of the lesion varies from host to host. For example, on watermelon, the lesions are small and circular, and on cucumber leaves, the lesions are large and angular. The main sign of downy mildew is the sporangiophore and the sporangia. These structures grow out of stomata on the abaxial side of the infected leaf. The many sporangia growing on the abaxial side of the leaf give the leaf a purple, gray, brown, fuzzy, or downy appearance. Sporangia are released and dispersed through the wind. It is thought that P. cubensis persists year-round in locations that do not experience frost. In the United States, it is suspected that each summer, downy mildew spreads from Florida up the East Coast. P. cubensis could also overwinter in plant tissue. When sporangia land on a new cucurbit leaf, there must be water present for their germination. Note the lemon shape of the sporangia and the papilla on one end. Each sporangia contain 5 to 15 biflagellate zoospores, which, when released from the sporangia, swim through the free water toward the leaf's stomata. When zoospores find a stomata on the leaf surface, they lose their flagella, encyst, and form a germ tube through the stomatal opening. The cenocytic hyphae of the pathogen grow through the intercellular spaces. They form haustoria inside the mesophyll cells by putting pressure on the cell and invaginating the cell membrane. This is where the pathogen absorbs host nutrients and transfers effectors into the host. This incubation period takes from 4 to 12 days. Sporangiophores emerge from stomata on the abaxial side of the leaf and chlorotic lesions form. Soon after, sporulation occurs. Sporangia form on the sterigmata, the tips of the dichotomously branched sporangiophores. The density of sporangia on infected leaves varies from host to host, but the general range is from 4,000 sporangia per square centimeter to 100,000 sporangia per square centimeter. The new sporangia can be blown away and can infect other plants. Therefore, this disease is polycyclic. So although P. cubensis reproduces asexually, it is able to disperse many propagules over long geographic distances, making it an especially devastating disease. Note that in this animation, the fungal structures are not made to scale, since each of the sporangia should be able to contain 5 to 15 zoospores. As time goes on, lesions can combine to form mass chlorosis and eventually necrosis. P. cubensis is an obligate parasite, so it will only benefit from the leaf when it is alive. There have been few reports of the sexual stage of P. cubensis, but it is rarely seen in nature, and its role in the epidemic has not been closely studied. Now I will show you the full animation.